This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas.
Imagine a forest at night. Or, maybe a really dense, quiet jungle.
Okay, I'm picturing it.
You can't see much, right?
Yeah.
But you just know that things are moving out there in the dark.
Oh, absolutely. It's that eerie feeling of being surrounded by hidden things.
Yeah, exactly. Now, take that exact feeling, that sense of something unseen shifting and changing just out of sight, and I want you to shrink it down to a microscopic scale.
Oh, wow. Okay.
Put it right inside the human body.
Well, that is a profoundly unsettling thought.
Right.
But, it's not just a metaphor. It's literal biological reality.
It really is.
Yeah.
And we are starting today's deep dive into the sources by talking about a very real ghost.
A ghost that caused a lot of panic, honestly.
Yeah, a microscopic phantom that basically haunted global health databases recently.
Yeah.
Officially, the World Health Organization designated it as a COVID-19 variant called BA.3.2.
Right.
But the scientists tracking it, they gave it a much more, evocative nickname. They called it the cicada.
Which is just brilliant branding, honestly, considering its life cycle.
It really is. For those of you listening, you probably remember the initial shockwave of the Omicron variant. It fundamentally changed the whole pandemic.
Oh, overnight. It was a complete paradigm shift.
Yeah, but what you might not know is that almost the second Omicron hit the global stage, it splintered.
Right. It didn't just stay one thing.
Exactly. It broke into three distinct lineages. There was BA.1, BA.2, and BA.3.
And we all know the first two.
Right. BA.1 and BA.2 went on to become, household names. They drove these massive, overwhelming waves of infection all around the world. But BA.3...
it just kind of vanished.
Yeah. It was an evolutionary dud. It couldn't compete with its siblings, so it just faded into the background noise of global genetic sequencing.
And the assumption among virologists was that it just hit an evolutionary dead end.
Just a bad roll of the dice.
Exactly. The virus rolled the genetic dice, got a bad hand, and the lineage basically died out.
Right. And that's what everyone thought, until, 2024.
That's when things got weird.
Very weird. Suddenly, out of nowhere, a descendant of that supposedly dead branch lights up on a sequencer, BA.3.2.
Just completely out of the blue.
Yeah. It had been evolving completely undetected for months, maybe even years.
Like a cicada.
Like a cicada.
You're rogue.
Burrowed deep underground, quietly shedding its skin, changing in the dark before finally digging its way back into the light.
And when it emerged, it really wasn't the same virus that went underground. It was heavily armored.
So how does that even happen? The prevailing theory I saw in our sources is that the cicada variant was likely incubating inside a single person.
Yes, that's the leading hypothesis, someone who was chronically infected.
Like an immunocompromised patient.
Exactly. For months or years, their body just couldn't clear the virus, so the virus just sat there using that person as a private training camp.
A private training camp? That's a wild way to think about it.
But it's true. It just sat there gathering dozens of new bizarre mutations.
Man, it really subverts everything we find comforting about modern medicine, doesn't it?
Oh, completely. We want diagnostics to be like an X-ray.
Right. Like a bone is broken or it isn't. Simple.
Exactly. But zoonotic disease, viruses that jump from animals to humans and viral evolution, this is a landscape of diagnostic muddy waters.
Muddy, fluid, chaotic, and mostly invisible.
Very much so.
Which brings us to the actual mission of our deep dive today. We are unpacking a stack of sources that try to map this invisible chaos.
It's a huge undertaking.
It is. We want to know, how do these pathogens shapeshift so effectively? How does a changing planet act as an accelerant for that shapeshifting?
Right, the macro drivers.
Yeah. And perhaps most incredibly, how are a group of scientists learning to build a kind of biological time machine to predict a virus's next move before it even makes it?
We are really looking at a remarkable synthesis of information today.
We really are.
Because we have to connect macro-level climate science and animal migrations all the way down to the microscopic machinery of viral RNA.
It's a massive scale.
It is. And the destination of this whole journey is a groundbreaking 2026 paper published in Nature Communications.
Oh, I can't wait to get to that one.
It's fascinating. A team of researchers basically figured out how to force a virus to show its evolutionary hand.
Okay, let's unpack this, because I quickly realized while reading through these sources that you can't understand a single microscopic mutation in a test tube without zooming all the way out first.
No, you need the context.
Right. We have to look at the macro engine driving disease emergence across the entire globe. And according to research from the Viral Emergence Research Initiative, they go by Verena, based out of Georgetown University. That engine is climate change.
Yes. Colin Carlson and Gregory Elbury led this research, and their predictive models are, well, they're paradigm shifting.
So what's the baseline premise here?
The premise is rooted in a reality we're already living. Earth is roughly 1.2 degrees Celsius warmer than pre-industrial levels.
Which, I'll admit, sometimes feels abstract, you know?
I hear that a lot. It sounds small.
Yeah. I mean, 1.2 degrees is literally the difference between me wearing a light sweater or just a t-shirt when I go outside.
Right, but on a planetary scale, the physics of that extra heat are violently rearranging the board.
Violently rearranging. Walk me through that. What does that actually look like for the animals?
Well, entire ecosystems are basically being evicted. Wild animals have what biologists call thermal niches.
Thermal niches, like a comfort zone.
Exactly. These are highly specific temperature and humidity bands where an animal's metabolism works perfectly.
Oh, I see.
It's where their food sources thrive, where their reproductive cycles naturally align with the seasons.
Okay, so it's their entire way of life.
Yes. And as the planet absorbs more heat, those thermal bands are physically moving.
Moving where?
They are sliding toward the poles, or they're creeping up to higher mountain elevations to escape the heat.
So the animals don't really have a choice. I mean, if the temperature band moves, you pack up your habitat and follow the weather, or you die.
That's the brutal reality. They are becoming climate refugees. And as they undertake these massive migrations, they're crossing borders they've never crossed before.
Right, because the map is changing.
Think about a species of deer or a rodent that has lived in a specific, isolated valley for 100,000 years. Suddenly, it's walking into a completely foreign forest.
And it's encountering other mammal species for the very first time in its evolutionary history.
Precisely. It's a global game of musical chairs.
But the music keeps speeding up.
Mm-hmm.
But let me challenge this slightly, just to play devil's advocate based on the sources.
Go for it.
Ecosystems aren't static museums, right? Animals have always migrated. Borders have always been porous to some extent.
They're over long time scales.
Right. So what makes this climate-driven migration so fundamentally different from, say, a wet market? Because we already know wet markets are the classic hotspots for viral spillover.
That is a really critical distinction to make, and Carlson's work actually addresses this directly by dissecting the mechanics of a spillover event.
Okay, how so?
Think about the conditions of a physical wildlife market. What do you see?
Well, you have high-stress environments, unsanitary conditions.
And highly unnatural proximity.
Right. You have animals stacked in wire cages, breathing on each other, animals that would never, ever cross paths in the wild.
And that unnatural proximity is the actual spark. Because a virus rarely jumps straight from a wild bat into a human to start a global pandemic.
Yeah, it needs a stepping stone, right?
Yes, an evolutionary stepping stone.
Yeah.
Take the 2002 SARS outbreak as an example. The virus originated in horseshoe bats.
Okay.
But humans don't generally interact with horseshoe bats in their daily lives.
So how did it get to us?
The virus needed to spill over into palm civets first. Those are small mammals that were sold in those physical markets.
Oh, I see. So it jumped to the civets, adapted inside them.
Optimized itself, yes, and then made the jump to humans. The market was a manufactured bridge.
Okay, I see where this is going with the climate model.
Right. What the Georgetown researchers are modeling is that climate change is taking that highly concentrated manufactured bridge and turning it into a global ambient reality.
Oh, man. So we don't need a crowded physical marketplace in a city center to create those unnatural combinations of stressed animals anymore.
Exactly. The shifting thermal niches mean the entire wilderness is becoming a borderless wet market.
A borderless wet market. That is a staggering way to visualize it.
Yeah.
The wilderness itself becomes the market.
And everywhere.
And the sources point out one specific type of animal that serves as the ultimate super spreader in this new borderless market.
Bats.
Bats.
They're the undisputed champions of viral sharing in these predictive models.
And reading this, it comes down to a very simple biological advantage, right? Flight.
It's that simple.
Because, I mean, if you are a migrating terrestrial mammal, like a badger or a deer, Your geographic range shift might only be a few miles a decade.
Yeah.
You are walking.
Slowly.
Yeah. But a bat can cross an entire continent in a season.
Their dispersal ability is completely unmatched among mammals. They travel vast distances.
And they're sampling diverse ecosystems along the way.
They interact with entirely naive animal populations, and they shed viruses the entire time they're traveling.
The sources actually provide a highly specific, very troubling hypothetical about this.
The Brazilian free-tailed bat.
Yes. Imagine its habitat in South America warms way beyond its thermal tolerance, so it begins migrating north.
Seeking cooler climates.
Right. So what happens when that bat eventually makes its way all the way into, say, Appalachia?
Well, the wildlife native to the Appalachian Mountains have zero evolutionary history with South American bat viruses.
None at all.
Their immune systems are a complete blank slate. They are sitting ducks.
I mean, it doesn't stop at the wildlife, which is the scariest part for us. The humans living in those regions are equally naive to those pathogens.
Precisely. The models project that by the year 2070, the new climate-shifted habitats of these migrating animals are going to disproportionately overlap with dense human settlements.
So we are looking at the emergence of just unprecedented cross-species transmission hotspot.
On a scale we've never seen.
And the geographic locations for these predicted hotspots are terrifying. We're talking equatorial Africa, South China, India, and Southeast Asia.
Southeast Asia is particularly alarming.
Why is that region so critical in the models?
Because it's already one of the most biodiverse regions on Earth for bat species specifically.
Okay, so you have the highest concentration of highly mobile, virus-carrying mammals.
Colliding directly with some of the fastest-growing, densest human populations on the planet.
It's a powder keg.
Yeah.
And we cannot stress enough to everyone listening that this is not a science fiction scenario for 2070.
No, it's not some distant hypothetical.
Right. The momentum of the climate system means that even if, say, all carbon emissions just magically cease today, decades of geographic range shifting are already locked in.
The engine is running. We can't turn it off overnight.
So the macro picture is clear. The board is moving, the pieces are colliding, and the opportunities for zoonotic spillover are skyrocketing.
Indisputable.
But wait. I have a question here. If this is happening everywhere, all the time, shouldn't our current epidemiological data be just screaming at us?
You would think so, right?
Yeah. Proving this mathematical link between a warmer day and a new disease outbreak must be, I don't know, incredibly complex if we aren't seeing it clearly yet.
It is notoriously difficult. And to understand why the data doesn't just hand us easy answers, we have to look at a fascinating preprint study by Trebsky and colleagues.
This was the one on the Medref server, right?
Yes. They set out to conduct a massive meta-analysis. They wanted to see just how widespread this climate sensitivity is across all zoonotic diseases.
And their conclusion was pretty definitive, wasn't it?
Oh, unequivocally, yes.
Yeah.
Diseases transmitted by vectors, so think mosquitoes carrying dengue or ticks carrying Lyme, and diseases transmitted directly from wildlife reservoirs are highly, highly sensitive to temperature, precipitation, and humidity changes.
Okay, so the link is real.
It's very real. But, and this is a massive but, Trebsky's team discovered that our current epidemiological data is just plagued by noise.
So, to use our earlier analogy, the diagnostic X-ray machine is broken.
Severely broken. They analyzed hundreds of studies and found profound methodological limitations.
Like what?
Well, for instance, over half the studies they reviewed didn't even report a clear sample size.
Wait, really? How do you publish without a clear sample size?
It happens more than you'd think in field biology, but the most insidious problem they identified was a consistent failure to control for spatial and temporal biases.
Okay, I want to pause on that. Spatial and temporal bias.
Yeah.
Because my gut reaction when I read that is that this is the classic streetlight effect.
Yes. The streetlight effect perfectly captures it.
Like the old joke about the guy looking for his lost keys at night under the streetlight. A cop asks him where he dropped them, and he points down the dark street and says, over there, but the light is better over here.
Exactly.
Are we really only finding viruses where it's convenient for us to look?
That is exactly what's happening. Think about how field biology actually works in practice. If you are a researcher trying to track bat viruses, where do you set up your traps?
I mean, I'm not hiking 50 miles into an untracked jungle if I don't have to.
Right. You probably set them up near roads, near your research outposts, or close to university campuses. Yeah. And temporally, you probably do it during the summer when the weather is cooperative and graduate students are available for field work.
Because they have time off from classes. That makes total sense. So the data we have reflects the habits of the human researchers, not just the habits of the viruses.
Precisely. And if your data is heavily skewed towards specific geographic locations and specific summer moths, it creates a massive temporal and spatial bias.
It's just noise.
Right. It becomes exceedingly difficult to untangle the real mathematical correlation between a 1-degree temperature rise and a disease outbreak, because the noise of human convenience is completely drowning out the actual biological signal.
Wow. This feels like a crucial pivot point for our deep dive today.
It is the turning point.
Because we know the macro engine of climate change is throwing all these species into a chaotic blender. We know the spillover events are increasing.
The physics guarantee it.
But field data, literally chasing animals through the woods, is too noisy, too biased, and frankly, too slow to predict what a virus will actually do when it finally jumps.
Field data is basically autopsy work.
Autopsy work.
Yeah, it's observing the aftermath of the chaos. If we want to move from observing the aftermath to actually predicting the future, we have to abandon the macro level entirely.
We have to zoom in.
We have to zoom all the way down to the atomic level. We have to understand the literal mechanical gears of how a virus adapts to a brand new host.
We have to look at the micro arms race.
Yeah, exactly.
Because when that Brazilian bat virus suddenly finds itself inside an Appalachian deer or, a human being, it doesn't just sit there politely. It has to figure out how to pick the locks of its new environment.
And the biological mechanics of that lockpicking are just mesmerizing.
So, to understand this lockpicking, our sources include a comprehensive review published in MDPI. This is by Jacob Warger and Silvana Gaudieri.
Yes, a fantastic overview of viral mechanics.
They map out the relentless microscopic arms race between a virus's need to adapt and a host's immune system trying to destroy it. Let's get into the mechanics of this shape-shifting.
Okay, let's break it down.
Because when we say evolution, we're really just talking about genetic typos, right? Mistakes.
At its core, yes.
Yeah.
Let's focus on RNA viruses, which is the family that includes Ebola, influenza, and SARS-CoV-2.
Okay, RNA viruses.
Their genetic instruction manual is written in RNA. When one of these viruses successfully breaches a human cell, it hijacks the cell's internal machinery to churn out millions of copies of itself.
It turns our cells into virus factories.
Right. And to execute this rapid copying process, the virus relies on an enzyme called an RNA-dependent RNA polymerase.
That's my full.
We usually just call it the RDRP.
RDRP. Okay, I always picture the RDRP as like a frantic typist.
A typist, I like that.
Yeah, so the virus hands this typist a manuscript and says, type this out 1,000,000 times as fast as you possibly can. But the typist doesn't have a backspace key.
They're just typing blind.
Right. They're just hammering the keys at 1,000 words a minute. And they're inevitably going to make typos. And whatever typos they make, they just stay on the page.
That is the perfect way to visualize it. And in biological terms, the vast majority of those typos are completely disastrous for the virus.
Oh, really? They hurt the virus.
We call them deleterious mutations. Say the typist accidentally deletes a crucial paragraph detailing how to build the viral envelope.
Then that copy is useless.
Exactly. That specific viral copy collapses and dies.
But every once in a while, the typist hits the wrong key and accidentally writes a better sentence.
Yes. A typo occurs that actually grants the virus a mechanical advantage.
Just purely by chance.
Pure chance. Maybe the shape of its surface protein changes just enough that a human antibody can no longer grab onto it.
So it slips by.
Right. That lucky mutant survives, replicates, and rapidly becomes the new dominant version of the virus. That sloppy copying is the engine of viral evolution.
Okay, I totally get that. But reading through the Warger and Gaudieri review, I hit a massive contradiction that I need you to explain to me.
What's the contradiction?
They note that coronaviruses are an exception to this rule. They say coronaviruses actually do have a backspace key.
Ah, yes. This is one of the most elegant and, frankly, devious features of the coronavirus family.
How does it work?
Well, unlike influenza or HIV, coronaviruses possess a specialized proofreading enzyme. It's a non-structural protein called NSP14.
NSP14.
And it functions as a 3' to 5' exonuclease.
Meaning it literally follows the frantic typist, looks for mismatched letters, cuts them out, and pastes the correct ones in.
Yes, it acts as an active spell checker.
That's incredible.
It really is. And this proofreader is why coronaviruses are able to maintain incredibly long genomes compared to other RNA viruses.
Because if they didn't have it, they'd make too many mistakes.
Exactly. If a standard RNA virus tried to carry as much genetic code as SARS-CoV-2 does, the sheer volume of uncorrected typos would trigger what we call a mutational meltdown.
The genome would just shatter under its own error rate.
Right, the proofreader stabilizes the virus so it can carry more instructions.
But this is where I'm deeply confused. If they have a dedicated spell checker, making sure the copies are accurate, how on earth did we get Alpha, Beta, Delta, and Omicron in the span of just 2 years?
It seems impossible, right?
Yeah. How did the cicada variant accumulate dozens of mutations in a single patient if the virus is actively fixing its own typos?
That paradox is the absolute crux of the arms race. It's a brilliant evolutionary strategy on the part of the virus.
So how does it pull it off?
The proofreader, NSP14, is highly active in protecting the main body of the viral genome.
Core instructions.
Right, the sections responsible for basic replication and structural integrity. The virus cannot afford typos there. But the virus essentially applies a totally different set of rules to one highly specific region.
Let me guess. The spike protein.
The spike protein.
The literal key the virus uses to jam into the locks on the outside of our cells.
Exactly. The virus allows the proofreader to be somewhat, well, lenient when it comes to the spike protein.
Lenient, like it just turns a blind eye to typos there.
Yes, and it has to. Spike protein is highly immunogenic.
Meaning what exactly?
It is the giant waving flag that our human immune system recognizes. Our bodies build customized antibodies designed to perfectly cap the tip of that specific spike shape, neutralizing the virus.
Oh, I see. So if the spell checker kept the spike protein perfectly identical, copy after copy, the virus would be committing suicide.
Exactly. Our immune system would memorize the shape once and destroy every subsequent infection effortlessly.
Wow. So the virus is forced into a deadly calculation.
Very deadly. The evolutionary advantage of changing the spike's shape to escape our antibodies vastly outweighs the risk of accumulating fatal typos in that region.
It's a trade-off.
A massive one. The virus intentionally tolerates a high mutation rate on its outer coat because it is literally the only way to survive the relentless adaptive pressure of the human immune system.
We build a better lock, they tolerate some genetic chaos to forge a new key.
We design a new vaccine, they forge a new key.
And that brings us right back to the cicada variant we talked about at the start.
Yes, the cicada is the ultimate manifestation of this exact arms race.
Right. Because that immunocompromised patient couldn't clear the infection, their body became a pressure cooker.
A closed-loop battleground.
The patient's weakened immune system would throw whatever antibodies it could muster at the virus. The virus would mutate its spike protein to dodge them. The immune system would recalibrate, try again, and the virus would mutate again.
A silent, months-long war of attrition.
And observing this war is fascinating. But as we concluded with the climate data earlier, observing things in the wild, whether it's inside a bat in a forest or inside a patient in a hospital bed, is just incredibly noisy.
Too much noise. There are too many uncontrolled variables.
Right, like what is the patient's exact antibody concentration? What other medications are they taking? You can't derive pure mathematical prediction from that kind of chaos.
No, you really can't. If we really want to understand the exact mechanics of how a virus chooses its shape, we have to strip away the noise of the real world.
We have to take control.
We have to force the virus into a highly controlled environment where we dictate the terms of survival.
And that profound leap from passive observation to deliberate, controlled experimentation is what brings us to the centerpiece of our deep dive today.
The 2026 paper.
Yes, published in Nature Communications. The paper is titled Stringent Selection Drives Convergence Toward Omicron-like SARS-CoV-2 Receptor Binding Motifs.
It's a brilliant piece of work.
Let's introduce the architects here. We're looking at lead authors Aviv Shoshani, Rujan Tian, and Miguel Padilla-Blanco.
And they conducted this research under the guidance of Gideon Schreiber at the Weizmann Institute of Science in Israel and Jiří Zaradnik at Charles University in Prague.
So what was their main goal?
This team set out to answer one of the most fundamental nagging questions of the entire pandemic.
Yeah.
What actually built Omicron?
Because structurally speaking, Omicron was bizarre. It was a complete anomaly.
It really was a massive, unprecedented evolutionary leap.
Right. Previous variants like Alpha or Delta, they had a handful of mutations on the spike protein. But Omicron arrived with dozens of alterations, heavily clustered right on the receptor binding domain, or RBD.
The RBD, yes.
And just to clarify, if the spike protein is a key, the RBD is the serrated edge of the key that actually slides into the pins of the lock. Right.
Perfect analogy. It's the part making physical contact with the human cell receptor, which we call ACE2.
Okay, so Omicron's RBD was heavily mutated. Now, the overwhelming consensus in the scientific community at the time was that Omicron looked like a freak because it was desperately trying to survive in a hostile world.
That was the running theory. By the time Omicron emerged, humanity had built up a massive wall of immunity through global vaccination campaigns and previous infection waves.
So the assumption was that immune evasion was the steering wheel. The virus mutated so heavily purely to hide from our antibodies.
It makes complete sense intuitively.
It does. But Shoshani, Tien, and their colleagues, they weren't satisfied with assumptions. They asked a brilliant, contrarian question.
What if we're wrong?
Yeah. What if immune evasion wasn't the main driver? What if something else entirely forced the virus into that shape?
And to test that hypothesis, they knew they had to isolate the variables.
How do you even do that?
They needed to see how the virus would evolve if they removed the human immune system from the equation entirely.
Just take it out completely.
So they built a high-throughput in vitro evolution system.
In vitro, meaning in glass.
And
In the lab.
yes. They use a technique called yeast surface display.
And reading about how this works felt like reading literal science fiction. I want to describe this setup because it's just a masterpiece of biological engineering.
It really is. Walk us through the arena.
Okay, imagine a microscopic gladiator arena. The scientists take ordinary yeast cells, like virtually identical to the stuff you buy in packets to bake sourdough bread.
Baking yeast, basically.
Yeah. But they genetically hack the yeast. They program the yeast's DNA so that as the yeast cell grows, it extrudes thousands of copies of the viral receptor binding domain directly onto its outer cell wall.
So the yeast cell becomes covered in tiny viral Velcro hooks.
Exactly. Viral Velcro.
Yeah.
So now you have millions of these fuzzy, weaponized yeast cells.
Gladiators.
The scientists drop them into a liquid solution. And floating in that liquid is the prize. Pure human ACE2 receptors. The biological lock.
But here is the trick.
Right. The researchers have attached a fluorescent molecular tag to the ACE2. It literally glows under a laser.
This is the absolute genius of the assay. The yeast cells tumble randomly in the liquid.
And if the viral Velcro on a yeast cell happens to successfully grab onto the glowing ACE2 receptor, it holds on tight.
And suddenly that entire yeast cell lights up like a microscopic Christmas tree.
I love that image. Next, they take this liquid soup of glowing and non-glowing cells and run it through a machine called a flow cytometer.
The bouncer.
The bouncer. This is essentially a hyperspeed laser-powered bouncer. It looks at millions of these yeast cells, one by one, in a fraction of a second.
It's incredibly fast.
If a cell is glowing brightly, meaning it's successfully grabbed the human receptor, the laser sorts it into a winner tube.
And if a cell remains dark,
Meaning its grip was weak or it failed entirely, it gets dumped into the trash.
it is a ruthless, hyper-accelerated simulation of natural selection.
And they don't stop there. They take the winners from tube A, artificially mutate their DNA slightly to mimic that frantic typist of viral replication.
They induce the typos artificially.
Exactly. They grow a fresh batch of yeast with those new mutations and throw them right back into the arena for round 2.
But you have to realize the logistics of running a tournament like this are terrifying from a lab perspective.
Why is that?
If you were running multiple lineages through multiple rounds, cross-contamination would ruin years of work.
Oh, because everything is microscopic.
Right. If a highly evolved super mutant clone from round 5 accidentally splashes into the petri dish for round 1, your entire evolutionary timeline is destroyed.
How do they control for that? How do you keep it straight?
That level of rigor is what elevated this paper into Nature Communications. Shoshani and Tien didn't rely on just keeping the tubes separate. They used a system of in situ barcodes.
In situ barcodes, what is that?
They inserted unique, non-functional sequences of DNA, literal molecular name tags, into the genetic instructions of the different viral lineages they were testing.
Like putting a tiny microchip on every single gladiator?
Exactly. They also employed different antibiotic resistance markers for different starting libraries.
So what's the benefit of that?
This meant that at the very end of the experiment, they could pull any winning clone, sequence its DNA, read the barcode, and know with absolute forensic certainty exactly which starting strain it came from and exactly which rounds of the arena it had survived.
That is bulletproof methodology. Okay, so the arena is built, the tracking is flawless, and the rules are set. And the most important rule of this arena is what's missing.
The immune system.
Right. By putting the viral protein on yeast and testing it against pure floating ACE2 receptors in a sterile tube, they completely eliminated the human immune system.
0 antibodies. 0 T cells.
They stripped away the noise of the arms race entirely. They isolated just one single evolutionary pressure, the raw mechanical ability of the virus to physically bind to the human cell and maintain its own physical stability.
Bind or be trashed by the laser.
And here is where the experiment splits into two divergent realities. The researchers didn't just run one homogeneous tournament. They forced the virus to evolve under two completely opposite conditions.
The two stringencies.
Yeah, they call them low stringency selection, or LSS, and high stringency selection, HSS.
The path of luxury and the path of starvation.
I love those terms. Let's look at the path of luxury first, the LSS. In the low stringency selection, the researchers just flooded the arena with glowing ACE2 receptors. The concentration was incredibly high.
There was plenty of food to go around.
And the evolutionary outcome was fascinating. When the environment is that forgiving, the virus gets lazy.
It really does. Survival requires very little effort.
The virus doesn't need to hone itself into a perfect, streamlined weapon. It can afford to accumulate a massive mess of sloppy random mutations.
Because almost anything it tries works well enough to grab an ACE2 receptor and survive the laser.
We see this clearly in the data. Under these luxurious conditions, a mutation called Q498H rapidly emerged and just dominated the population.
Q498H, meaning at position 498 on the spike protein, the amino acid glutamine, represented by the letter Q, mutated into histidine, represented by H.
Now, why is Q498 interesting? Because historically, virologists see this specific mutation emerge when the virus is forced to adapt to infect mice.
Ah, mouse adaptation.
Right. It popped up in older, less rigorous lab experiments. But crucially, Q498H never became a major player in the real-world human pandemic. Yet in the luxury lab arena, it flourished.
The researchers actually pushed this luxury concept to the absolute extreme with a secondary experiment.
Oh, right, the superbinder.
Yes. They took a highly optimized, heavily mutated variant of the receptor binding domain that they had engineered previously. They called it RBD-V48.
This thing was a monster. Right?
It was. It already possessed a binding affinity 50 times stronger than the original Wuhan strain.
50 times.
So they dropped this superbinder into the luxury arena.
Now, you would assume it would just refine itself into ultimate perfection, right?
You'd think so. Instead, it completely lost its edge. It absorbed bizarre, rare mutations. Mutations like A348P or K386E.
Do those actually do anything?
No, they are alterations that provide almost zero biological advantage in binding. But because the virus already had such a massive, unneeded surplus of binding strength, it could tolerate these useless mutations without being penalized by the flow cytometer.
I love this concept. It's like a billionaire who just stops balancing their checkbook.
That's a great way to put it.
Right. They have so much excess capital that they start making terrible investments, buying weird art, funding doomed startups because they can afford to lose the money without going bankrupt.
The virus had a billionaire budget of binding affinity.
So it just wandered aimlessly through the evolutionary landscape. And the key takeaway here is that under low stringency selection, the luxury path, the virus never converged on anything that looked remotely like Omicron.
No. But then the researchers turned up the dial. They initiated the high stringency selection, HSS.
The path of starvation.
They drastically reduced the concentration of ACE2 receptors in the liquid.
Suddenly, the arena was barren.
There was hardly any target available. The viral clones had to fight viciously for survival.
Only the absolute tightest, strongest, most desperate binders would capture enough of the rare glowing ACE2 to trigger the laser and avoid the trash bin.
And the results of this high-pressure tournament are the beating heart of this entire Nature Communications paper.
They really are.
The results were staggering. In just 2 rounds of mutation and selection, 2 brief generations in the arena, the virus rapidly, identically, and repeatedly converged on a very specific set of mutations.
Regardless of what genetic background they started with, the intense pressure forced the virus to select for mutations N440K, S477N, T478K, Q498R, and N501Y.
Those specific amino acid substitutions represent the exact genetic fingerprint of the Omicron variant.
It is mind-blowing. In just 2 rounds of pure intense pressure to bind to the human lock, the virus in a glass tube perfectly recreated the hallmark mutations that defined a global pandemic wave in the real world.
It's incredible validation of their system, and the researchers highlighted a profound synergistic magic that happened under this pressure, specifically between 2 of those mutations, Q498R and N501Y.
Explain that synergy. What actually happens chemically when they combine?
Well, when a virus acquires the N501Y mutation alone, its physical shape shifts slightly, allowing it to bind a bit better to the ACE2 receptor.
Okay, an incremental improvement.
And when it acquires Q498R alone, it also binds a bit better. But when the virus was forced to compete in the starvation arena, it essentially figured out a cheat code.
Cheat code.
It learned that combining those two specific mutations didn't just add their strengths together, it multiplied them.
Oh, wow.
They interacted with each other structurally to perfectly mold the viral key to the contours of the human lock.
So the clones that survived the high stringency selection didn't just visually resemble Omicron on a sequencer. They functioned like elite, Olympic-level athletes.
They were the peak performers.
The researchers actually measured their grip strength. They found that the HSS clones bound 10 times tighter to human ACE2 than the clones from the luxury arena. We are talking about binding affinities in the 0.4 to 1.5 nanomolar range.
To contextualize that, for a non-chemist, affinity is measured inversely.
Inverse, meaning lower is better.
Exactly. A smaller nanomolar number means a much tighter bond. A bond in the subnanomolar range is terrifyingly efficient.
Meaning what in practical terms?
It means that if a single viral particle even brushes against a human cell receptor, it is locking on instantly and permanently.
Okay, so this is just an incredible piece of laboratory wizardry. But if we step back and ask the big question, what does this test tube tournament actually mean for our understanding of the real-world pandemic?
This is where Shoshani and Tian drop a philosophical bomb on the virology community.
A total paradigm shift.
It forces a complete rewrite of the narrative we've accepted for the last 3 years.
Let's get into it.
If we connect this finding to the bigger picture, remember the core assumption we started with. The world believed Omicron looked like a heavily mutated freak, primarily because it was twisting itself into knots to evade our vaccines.
Right, we believed immune evasion was the architect of Omicron.
But look at the gladiator arena. In the high-stringency lab experiment, there were no vaccines.
There were no antibodies at all.
Zero evolutionary pressure to hide from an immune system. The only pressure was a mechanical requirement to bind flawlessly to human ACE2.
And yet, the virus evolved into Omicron anyway.
Exactly.
Wait, I have to stop and push back on this because it feels completely counterintuitive.
I know, it's hard to wrap your head around.
Are the researchers claiming that Omicron's incredible ability to evade vaccines was just a happy accident? That the virus wasn't trying to hide at all? That's really hard to believe, given how easily it reinfected people.
They aren't saying immune evasion is a total accident, but they are proving that immune evasion was not the primary driver, selecting those specific hallmark mutations.
Okay, how do they prove that?
The paper clarifies this beautifully by looking at the fate of a famous mutation called E484K.
E484K. I remember reading about that one during the beta variant wave.
Yes.
It's notorious for being an incredible invisibility cloak.
Yes, it radically changes the shape of the spike so our antibodies just slide right off it.
Exactly. It's a masterclass in immune evasion.
Yeah.
But guess what happened to E484K in the high stringency arena?
Let me guess. The laser bouncer threw it in the trash.
It was overwhelmingly selected against.
Why would it throw away such a good defense?
Because while E484K is a fantastic invisibility cloak, that structural change actively weakens the virus's ability to bind tightly to the ACE2 lock.
Oh, so it sacrifices grip for stealth.
Right, and in a pure starvation-level fight for binding survival, E484K is a fatal liability.
That makes total sense. So what did the virus do instead? If it couldn't use E484K to hide, how did it survive?
It found a compromise. Under extreme binding pressure, the virus selected a different mutation at that exact same coordinate, E484A.
E484A, so it swapped a letter.
By swapping to alanine, represented by A instead of lysine K, the virus managed to keep its binding strength incredibly high, while still altering the surface charge just enough to slip past some antibodies.
It prioritized the mechanical grip on the human cell above all else and just took whatever marginal immune escape it could get without sacrificing that grip.
The grand conclusion here is profound. Omicron isn't just an immune-evading mutant that accidentally got good at spreading.
No, it's something much more purposeful.
Omicron is the ultimate, optimized, humanized binding machine. It is the virus perfectly and ruthlessly molding itself to the physical reality of human biology.
And the immune evasion we experience in the real world is largely a secondary byproduct of this
A
optimization.
byproduct.
Yes. As the virus relentlessly shifts its shape to fit our cellular locks more perfectly, it naturally alters the surface profile that our old antibodies were designed to recognize.
This balance between pure binding optimization and real-world survival is so delicate. And to really illustrate how complex this is, the researchers investigated a massive anomaly in their data.
The S375F anomaly.
Yes, a mutation called S375F.
S375S is a fascinating biological mystery. In the real world, the S375F mutation is practically mandatory for Omicron. Every single Omicron lineage circulating globally has it.
But when Shoshani Tian and the team forcibly engineered a virus with the S375F mutation and dropped it into their yeast arena,
What happened?
the yeast couldn't handle it. The clones possessing S375F performed terribly and were rapidly eliminated from the gene pool.
But why, if it's everywhere in the wild?
They discovered that S375F is structurally destabilizing. It alters the folding of the protein in a way that makes the entire spike wobbly.
Wobbly.
Yeah, in the pure, isolated environment of the yeast display, where raw stability and binding strength are the only metrics for survival. S375F is a fatal flaw.
So here's the million-dollar question. If it's a fatal flaw in the lab, why is it wildly successful in every Omicron virus in the wild?
Right, that contradiction tells scientists something deeply important.
It means S375F must provide some other mysterious, overwhelming fitness advantage in a living, breathing human body that completely overrides its destabilizing wobble.
What kind of advantage could possibly make a wobbly key worth keeping?
The leading hypothesis involves the physical posture of the spike protein itself. The spike protein isn't static, it can actually hinge open and closed.
Like a physical movement.
Yes. Think of it like a switchblade. If the receptor binding domain is always flipped out and exposed, it is very easy for circulating antibodies to spot it and attack it.
But if you keep the blade folded in the handle, you're hidden.
Precisely. The theory is that S375F acts like a faulty spring mechanism. It destabilizes the protein just enough to keep the spike in a down or closed conformation for longer periods of time.
So the virus sacrifices a bit of structural integrity to keep its most vulnerable parts hidden from the immune system until the exact microsecond it bumps against an ACE2 receptor and needs to strike.
Exactly. So in the wild, the evolutionary advantage of hiding from a furious immune system outweighs the mechanical wobble.
But in the lab, where there are no antibodies to hide from, the wobble just gets you tossed in the trash.
This perfectly demonstrates how the yeast arena acts like a prism, separating the pure pressure of binding from the messy pressure of the immune system.
That is such a cool way to look at it. A prism.
Yeah.
And once this Weissman Institute team realized the power of the prism they had built, they didn't stop with COVID-19.
No, they pushed it further.
They realized they had constructed a predictive engine. They called it the Phenotypic Selection Inference Framework.
Catchy name.
And they decided to test its predictive power on a ghost from the past.
They brought out the original SARS-CoV-1 virus.
The pathogen responsible for the 2002 outbreak.
SARS-1 is a really fascinating comparison point. Clinically, it was much deadlier than SARS-CoV-2, with a significantly higher fatality rate.
But it didn't trigger a multi-year global pandemic.
Because it didn't spread as easily. Its spike protein was clunky. It lacked that terrifying sub-nanomolar grip on the human ACE2 receptor.
So the researchers take the clunky SARS-1 spike protein, attach it to the yeast, and throw it into the high-stringency starvation arena.
Just see what happens under pressure.
Exactly. They want to see what it would take for this older, deadlier virus to learn how to bind like Omicron. They hit fast forward on its evolution.
The results of that simulation are equally terrifying and empowering.
Terrifying.
Because it only took one single mutation in the SARS-1 receptor binding domain to completely close the affinity gap.
One typo.
One specific change, a mutation called Y442S.
So just swapping a tyrosine for a serine.
By simply swapping a tyrosine Y for a serine S at position 442, the shape of the interface loop altered just enough to perfectly accommodate the human lock.
Wow.
That single mutation transformed the old clunky virus into a highly optimized pandemic-capable binder.
It is terrifying because it illustrates how thin the barrier is.
Yeah.
These older, deadlier coronaviruses are lurking in bat populations right now, and they might only be one genetic typo away from acquiring the transmissibility of Omicron.
Exactly. But the empowerment comes from the knowledge.
The wanted posters.
Yes. By running these in vitro simulations, we are no longer flying blind. Shoshani and Tien have basically handed the global health community a stack of biological wanted posters.
Right. We now have the exact genetic sequence of a future threat. Which brings us full circle back to the macro engine of climate change we discussed at the top of the show.
The borderless wet market.
As the planet warms, as the bats migrate into Appalachia or new parts of Southeast Asia, scientists are out there swabbing them and sequencing the viruses they carry.
And now, when a field researcher sequences a wild bat coronavirus, they don't have to guess if it's dangerous.
They run the sequence against the wanted poster.
If they find a virus carrying the Y442S mutation or the Q498R and N501Y combo, the red alarms go off immediately. We know its pandemic potential before it ever comes into contact with the human lung.
But identifying the threat is only half the battle, right?
Sadly, yes.
Knowing the train is coming doesn't help if you can't get off the tracks. If viruses are this incredibly good at shapeshifting, if a test tube can prove they only need 2 generations to mutate their spike proteins into a masterpiece of human adaptation, how do we actually defend ourselves?
We can't keep playing a global game of whack-a-mole.
Exactly. Trying to invent and distribute a new variant-specific vaccine every 6 months. The virus moves too fast.
Escaping that cycle of whack-a-mole is the ultimate holy grail of modern immunology.
And to understand how we fight back, your sources point us toward groundbreaking active research happening at the Loyola Institute for Immunology.
Spearheaded by scientists Alba Grifoni and Alessandra Sette.
Their goal is to develop a pan-coronavirus vaccine, a universal shield.
Which sounds impossible.
Structurally, how is that even possible? If the spike protein, the primary target of all our current vaccines, is constantly mutating its shape to escape our defenses, how do you make one single vaccine to fight a shape-shifting family of viruses?
You have to fundamentally change your target.
Change it to what?
You have to look past the spike. Think back to the proofreading enzyme we discussed earlier, NSP14.
The lenient spell checker.
The virus allows the spike to mutate wildly, but it fiercely protects the rest of its genetic code. There are internal proteins, internal machinery, and even the stem helix structure at the very base of the spike that the virus simply cannot afford to change.
If the frantic typist makes a mistake in those protected areas, the virus breaks.
Exactly. We call these areas highly conserved regions. Because they are mechanically essential, they remain virtually identical across the entire coronavirus family tree.
Oh, I see.
The genetic sequence for these internal structures is practically the same in a mild common cold coronavirus, in the deadly 2002 SARS-1 virus, and in every single variant of SARS-CoV-2. They are the universal Achilles heel.
So why don't our current vaccines just target those conserved regions? Why do we obsess over the spike if it just keeps changing?
Because of the limitations of antibodies. Antibodies are large, Y-shaped proteins. They operate in the bloodstream and outside of cells.
Okay, they're on patrol.
They are incredibly good at grabbing the flashy, exposed, outward-facing parts of a virus, like the tip of the spike, but they're physically too large and clumsy to penetrate the viral envelope or reach down into the hidden, conserved base of the spike.
So the antibodies can't reach the Achilles heel.
No. To attack the conserved regions, we have to harness an entirely different branch of our immune system. We have to activate the T cells.
T cells, the precision assassins of the immune system.
While antibodies try to block a virus from entering a cell in the first place, T cells have a different job. They constantly patrol the body, scanning the surface of our own cells.
Looking for trouble.
When a virus manages to infect a cell, the human cell chops up pieces of the virus's internal conserved proteins and displays them on its surface, like a distress flag.
Signaling for help.
T cells are expertly calibrated to recognize those specific internal distress flags. When they see one, they immediately destroy the infected cell, halting the virus's ability to replicate and spread.
So the strategy at the Loyola Institute, Grifoni and Sette, are painstakingly mapping the entire viral proteome to identify exactly which of these conserved internal pieces trigger the strongest T cell responses.
Yes. They are building vaccines designed not to induce antibodies against a shape-shifting spike, but to train robust cross-reactive T-cell memory against the unchanging backbone of the virus.
Wow. If you successfully train the T cells to recognize that hidden core, it renders the shape-shifting spike completely irrelevant.
It wouldn't matter at all.
It wouldn't matter if the virus evolves into an Omicron or a cicada or a completely novel bat virus we've never seen before. The moment it enters a cell and tries to replicate, the T cells will recognize the conserved architecture and terminate the infection.
A true pan-coronavirus vaccine could protect humanity against variants that haven't even evolved yet.
It represents a monumental shift in our approach to infectious disease. We are moving from a reactive posture, waiting for a spillover, suffering through a wave, and scrambling to formulate a countermeasure to a proactive, predictive posture.
By understanding the raw mechanics of viral binding and aiming our defenses at the virus's unchanging heart, we are learning to finally read the enemy's playbook.
It's an incredible synthesis of scientific disciplines. Let's trace the massive journey we've been on today. We started at the macro level, looking at a planet warming by 1.2 degrees.
We saw how shifting thermal niches are forcing bats and other wildlife into unprecedented migrations.
Turning global ecosystems into a chaotic, borderless wet market of viral sharing.
Then we zoomed into the micro level to understand the RNA arms race.
We saw how viruses utilize a proofreading enzyme to protect their core stability, while intentionally permitting their spike proteins to mutate furiously to evade human immune responses.
We followed the Weizmann Institute team as they captured that microscopic machinery
And subjected it to the brutal high-pressure environment of a yeast display gladiator arena. And they proved that we don't have to wait blindly for nature to act.
By applying pure, isolated mechanical pressure for human receptor binding, we can fast forward a virus's evolution in a test tube.
We can watch it converge, identically and repeatedly, into a pandemic-capable form in just 2 generations.
They proved that a variant like Omicron wasn't just a random assortment of immune-evading errors. It was a highly predictable, stringently selected masterpiece of human adaptation.
And finally, armed with the knowledge of how a virus changes its outer coat, we looked at how researchers are learning to aim our vaccines past the spike.
Training our T cells to attack the highly conserved, unchanging regions of the viral family tree to create universal pan-coronavirus defenses.
It is a profound testament to human ingenuity. The diagnostic muddy waters are finally beginning to clear.
We're finally getting an X-ray of the future.
Which leaves me with a final lingering thought for you to chew on as we wrap up today's deep dive.
I love a good lingering thought.
We've talked about putting SARS-CoV-1 and SARS-CoV-2 into these in vitro arenas to map their optimal binding pathways. But if we can take any virus, put it in a test tube, and fast forward its evolution to predict exactly what it will look like years from now.
Are we approaching a future where we systematically map the future history of every zoonotic virus currently migrating in the wild?
Exactly. A catalog of every potential pandemic before it happens.
It's theoretically possible now.
If we know exactly what a bat virus in Appalachia needs to do to perfectly infect a human cell, could we eventually use these T-cell-driven pan-virus platforms to proactively vaccinate populations against a pandemic variant that does not even exist yet in nature?
It sounds like science fiction, but the sources we review today prove that it is the active, tangible frontier of immunological research.
The microscopic cicada variant might have surprised us by hiding underground and mutating in the dark.
It won't be the last to try.
No, it won't. But thanks to the relentless predictive modeling of climate scientists, the ingenuity of lab researchers building evolutionary arenas, and the foresight of immunologists mapping universal vaccines, the next time a virus tries to emerge from the dark, we won't be caught off guard.
We'll already be waiting for it.
We'll already be waiting for it with the lights on. Thank you for joining us on this deep dive into the source material. We will catch you next time. Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.